You know, right now, inside your body, there are literally trillions of secret messages being sent back and forth. And no, I'm not talking about nerve impulses. We're talking about these incredible, microscopic packages that might just hold the key to the future of medicine.
So let me ask you this. What if I told you that a single blood test could spot something like cancer or Alzheimer's, not just early, but years before you ever felt a single symptom? It sounds like science fiction, doesn't it? Well, it's the very real promise of a technology that revolves around something called an exosome.
So what on earth is an exosome? Well, the best way to think about it is like your body's own microscopic postal service. It's this teeny tiny virus-sized package that pretty much every single cell in your body releases to send out critical messages and materials to other cells.
Okay, so if our cells are sending out mail, the most important question is pretty obvious, right? What's actually in these packages? What's written in the letter? Let's take a look.
And what's packed inside is, well, it's just incredible. These little vesicles are carrying a super rich cargo. We're talking proteins that act like ID tags, messenger RNA that holds genetic instructions, even little bits of DNA. It's basically a perfect molecular snapshot of the exact cell that sent it out.
And here's the absolute game changer. Research has confirmed that the stuff inside an exosome is a highly accurate reflection of its parent cell. So they're like tiny little informants. If a cell starts to become cancerous, guess what? The exosomes it sends out will carry the molecular signs of that cancer, basically tattling on their sick home cell to the rest of the body.
You can probably see where this is going. This ability to essentially spy on ourselves from a distance, well, that is exactly why exosomes are being called medicine's next great frontier. The potential here is just staggering. And this all leads us to the holy grail concept of a liquid biopsy. I mean, imagine this. Instead of a painful, invasive surgical biopsy to get tissue, your doctor could just take a simple blood sample. From that, they could isolate the exosomes and literally read the messages inside. It would be a totally non-invasive way to find disease, track how it's progressing, and even check if treatments are working, all from one blood draw.
And let me be clear, this isn't just some theory stuck in a lab somewhere. It's happening right now. There are dozens of clinical trials underway actively testing exosome-based diagnostics and therapies for, well, almost everything from cancer to heart disease to Alzheimer's. This is being tested in real patients as we speak. We're even seeing actual drugs in the development pipeline. Just look at ExoFlow. It's already in late-stage phase 3 trials, using exosomes to treat severe lung inflammation. Others are targeting serious burns and a whole host of other conditions. This tech isn't some distant dream anymore. It's getting closer and closer to our clinics every day.
And you knew there had to be a but, right? With all this amazing promise, there is a really significant challenge, a major reality check for the whole field. And it all comes down to what scientists are calling the purity problem.
So here's the core issue. To read those all-important messages, you first have to grab the exosomes out of the blood and separate them from, well, all the other junk that's floating around in there. And as of right now, there's no single, universally agreed upon, gold standard method for doing that cleanly and reliably.
In fact, get this, over 80% of researchers are using one main method to do this, called ultracentrifugation. And the thing is, it's a method that is known to have some pretty serious drawbacks, including the risk of actually damaging the very exosomes they're trying to study.
To really see why this is so tricky, let's just quickly compare two common methods. So, ultracentrifugation, that's like spinning a sample at unbelievable speeds to separate things by weight. It's fast, sure, but you often end up with a lot of unwanted proteins mixed in, low purity. On the other hand, you have something like size exclusion chromatography. Think of that as a really, really fine-tooth sieve. It gives you much higher purity, but you get a smaller sample at the end. It's this constant trade-off, and depending on which method a lab uses, they can get wildly different results.
And the researchers themselves put it pretty bluntly. They say that isolated exosome samples are often contaminated. It's like trying to find one critical, life-saving letter that's been thrown into a massive pile of junk mail. How can you be sure you've got the right message? This contamination issue is really the single biggest bottleneck holding the entire field back.
But of course, the story doesn't end there in frustration. Faced with this huge challenge, scientists and engineers are doing what they always do best. They are innovating. They are building a much, much better toolbox to solve this purity problem once and for all.
And the innovation here is just wild. We're seeing things like microfluidic chips. We're talking labs on a chip that can sort these exosomes with incredible precision. Scientists are also using powerful machine learning algorithms to sift through mountains of data and pinpoint the exact molecular signals of disease. It is a full-on technological assault to crack this problem.
So, what's the point of all this new tech? Well, the ultimate goal of all these new tools is to achieve one thing: consistency. To get reproducible, reliable results that a doctor can trust every single time. That is the key that's going to unlock the door to widespread clinical use and finally bring the promise of liquid biopsies to all of us.
So where does that leave us? We know these tiny messengers are carrying the secrets to our health, floating around in our bloodstream. The science is moving forward at an absolutely incredible pace, but the challenges are very real. The race is on to build the right tools, the right key, to unlock this new era of medicine. The only question left is, how quickly can we learn to read what our own bodies are already trying to tell us?